Hello, everyone. In the series of next generation sequencing lectures, we have discussed that short read data has read lengths of 36 base pair, 50 base pair, 75 base pair, 150 base pair, 250 and 300. And the long read data can have a read length from 15 to 25 KB and 100 KB. This short read data is generated by Illumina platform, whereas the long read data 15 to 25 KB can be generated by PacBio, and Nanopore, Oxford Nanopore technology can generate 15 KB to 25 KB, can also generate 100 KB fragments. And this is called as ONT, Oxford Nanopore technology. With this background, we'll have to now understand which reads have to be generated for what. Which reads have to be generated for what. So this lecture deals with this topic, and to understand this topic, we'll have to understand the classification that I gave you at the bottom. Now, NGS mainly involves DNA sequencing and RNA sequencing. And DNA sequencing is for variant calling, and DNA sequencing can also be used for assembly. RNA sequencing is used for mRNA-seq, whole transcriptome, and microRNA-seq. And this RNA-seq can also sometimes be used to assemble the RNA genome. Now we'll have a look at the read length that is needed for each one of these sequencings. For variant calling, when you talk about variant calling, we are talking about calling the SNPs. And this needs around 2 into 150 base pair read length. That means paired end data of 150 base pair read length is required for variant calling and to generate SNP data. When we talk about assembly is nothing but assembling the entire genome. In the eukaryotes, it's around 3 into 10 to the power of 9 base pairs in length, the total genome. For this, it is better to have long reads. Therefore, it is better to have 15 KB to 25 KB reads. And you can assemble the genome. However, people along with 15 KB to 25 KB reads also use 100 KB reads to complete the assembly. And such kind of assemblies can be now called as T to T assemblies. It means telomere to telomere assemblies. However, for assembling this entire genome, besides having these read lengths, it is also important to have bionano data and high-C Illumina data. High-C Illumina data. Now, for RNA sequencing, your RNA inside the cell is dominated by your rRNA. RNA also includes mRNA, which will normally be sequenced to find out the differentially expressed mRNA under different conditions of your study. And this mRNA codes for your proteins. Besides this, you have LNC RNA, which can directly affect your mRNA and affect the gene expression. And generally, we can do a co-expression analysis to find out the effect of LNC and mRNA together. Then we have microRNA. This microRNA, normally they bind to the 3' UTR of your transcript. For example, if this is your transcript, 5' to 3', this mRNA binds to the 3' UTR of your transcript. Because of the binding of this miRNA to your 3' UTR of the transcript, the ribosome that is coming and translating your transcript drops off. And this transcript is not translated into a protein. So this is how microRNA influence the gene expression. And finally, you have circular RNA. You have circular RNA, which will sponge your microRNA and prevent your microRNA to bind to your 3' UTR. Having got this background, we now get back to RNA sequencing. And when mRNA sequencing is done, here you are only looking at sequencing the mRNA. So the RNA that is isolated, is separated by using oligo-DT probes, because all your RNA contains poly-A tail, and so you isolate your mRNA from your entire transcriptome, and this mRNA is separated, and this mRNA is then fragmented and then sequenced. That is the fundamental here. So when you sequence this, the ideal length, read length for this sequencing is 2 into 75 base pair. Whereas here, when you go for whole transcriptome, here under the whole transcriptome, you aim at sequencing your mRNA, you aim at sequencing your LNC RNA, and also your circular RNA. So here, the RNA that is taken is depleted of the rRNAs, means your rRNAs are removed. And this process is called as ribodepletion. This process is called as ribodepletion. And once the rRNAs are removed, this RNA that is effectively got after the removal is again fragmented and sequenced. And the ideal length for this sequencing is 2 into 150 base pair. 2 into 150 base pair. Now, for your miRNA-seq, to isolate your microRNA, here you use a specialized kit, isolate your microRNA, and then sequence your microRNA. And this microRNA is normally sequenced by using read lengths of either 1 into 36 base pair or 1 into 50 base pair. And finally, sometimes you have a job of assembling the RNA genome in the case of RNA viruses. For this, you use 2 into 300 base pair sequencing. Nowadays, more often than not, instead of 2 into 300, people started to move towards 15 kb fragments, that is 15 kb to 25 kb PacBio reads. And this is what people are doing nowadays for RNA genome assembly. Thank you.